Hi, I'm Professor Chakraborty, and today's lecture is on neurophysiology, one of the very important topics of this course. We need to understand some basic information is that the communication of information between neurons, that is between one neuron and another neuron, or between neurons and muscles or peripheral organs, require signals to travel long distances. This, for example, in this cartoon, the person actually stepped into this nail, and the pain receptors took that information through the peripheral nervous system, which are the spinal nerves, to the spinal cord, where spinal reflex took place. And as a result, the person removed their foot immediately. This entire process happened in milliseconds, perhaps, without the involvement of even brain. And eventually, the brain did get involved, and the persons got into the pain receptors, and you can see tears coming out of his eyes. So this phenomenon is happening every single day. You touch an oven, someone pinches you, and so many such activities are happening in our day-to-day lives. And that's what we are going to learn in here. How does this happen? How does this entire process takes place? So, in order to learn about this entire process, what we are going to learn, or we are going to understand, the four questions which will help us to understand the entire chapter very well. Number one, why is the membrane potential inside a neuron negative and outside positive? Why is the resting membrane potential negative 70 millivolt? What is equilibrium potential? And what is graded and action potential? We'll start with the first one, which is what is membrane potential, of course. And once we know the membrane potential, why is the inside of the membrane negative and outside positive? So, as we all know, that neurons are electrically excitable. That's a characteristic of a neuron. The membranes are electrically excitable due to a voltage difference. What I mean by voltage differences is that there is difference in the distribution of ions across the membrane. So if you look at this diagram, this is the membrane, it's a phospholipid bilayer, and there are ions outside and there are ions inside. Sodium is higher outside, potassium is higher inside, and we learn why that is. And you can see that this difference in the distribution of ions causes what is called the transmembrane potential or membrane potential. So the transmembrane potential is the electrical voltage difference across the cell's plasma membrane. So the cell's electrical signal, there are two types. One is known as the graded potential, the other is known as the action potential. And we'll learn at length about both of them as we go along. These movement of ions, sodium, potassium in particular, causes what we call the movement of stimulus. So, what is the membrane potential and how is it created? So we learned what is membrane potential. And now we are learning how this membrane potential is created. And then we are learning why is the inside of the membrane, which is ICF, negative and outside ECF positive. There are particularly two ions that we are going to discuss mainly. One is the sodium ion, the other is the potassium ion. And there are three factors or three resources that are involved in this entire process. Number one is the sodium potassium pump, which actively transports three sodium ions from inside the cell to outside and two potassium ions from outside to inside. So if you consider a cell and a membrane which is not semi-permeable, so it's permeable to all the ions equally, an imaginary and hypothesis we are thinking about, then sodium, potassium, it should have equal distribution inside and outside of the cell, right? But it does, for example, it does, but there is a pump, which is sodium potassium pump. What it does, it transfers three sodium ions outside and brings in two sodium potassium ions inside. So let's look at the figure in this case. You will see that this is the sodium potassium pump. And this plays a very significant role in the membrane potential because it's the initiation of the membrane potential. This is a sodium potassium pump which uses ATP, which is the main energy source. A ton of ATP it uses. And what it does, it takes up three potassium ions and puts it outside of the membrane, which is ECF, extracellular fluid, and brings in two potassium ions inside. So 3:2. So outside you can see there are now more sodium ions, and inside there are more potassium ions with the help of these sodium potassium pump. So more sodium ions outside, which is positively charged, less potassium ion, which is also positively charged. But since sodium is more outside than potassium, there is a slight negative charge created in here. This is the initiation process, okay? This has initiated the difference in the ionic distribution, which is a transmembrane potential that we just learned, right? So this is the initiation. Maybe think about it minus -5 millivolt, for example, okay? So this is the first part. So if we go back to the slide, you will see apart from the sodium potassium pump, there are leak channels. Okay, so what does the leak channel do? The leak channels are present in the semi-permeable membrane, which give easy access to sodium and potassium. They can, they just let sodium and potassium to move freely. But what did we learn? The sodium is higher outside, potassium is higher inside. So what will happen is through these leak channels, sodium will move inside and potassium will move outside, right? So it will try to, create a balance. But just to make things more complicated, of course, what happens is the potassium leak channels, it's much more permeable to potassium ions than the sodium leak channel. So tons of potassium ions will go outside of this plasma membrane. As a result, what will happen is, since more potassium ions have gone outside, which is positive ions, this creates a further negative millivolt or transmembrane potential, which is nothing but -70 millivolt, which is a resting membrane potential. So the resting membrane potential of a neuron is -70 millivolt. So again, there are two factors that we have considered so far. One is sodium potassium ATPase, which requires ATP. And remember, most of the ATP in the brain is used by the sodium potassium ATPase to move these ions. So what it does, it removes three sodium ions from inside to outside and two potassium ions from outside to inside. And the leak channels are present, which in turn causes potassium ions to move outside, and leak channels help sodium to come inside. But the potassium leak channel is much more permeable to potassium. As a result, many potassium ions will leak outside. So this is the second reason why the negative 70 millivolt goes into negative 5 millivolt goes into negative 70 millivolt or around there in order to form the resting membrane potential. There's another factor, which is several anions are present inside the cell, which are produced by the cell. These anions, which are present in it, this is anions are A negative charges. These are large molecules and are unable to move out through these channels. As a result, they remain inside the cell and causes the negativity of the intracellular fluid. So all these three factors together contribute to the negative 70 millivolt, which is a resting membrane potential of the neuron. So from here, I'm going back one slide again so that we understand. We learned about sodium potassium pump, we learned about leak channels. Now we are going to learn about the next part, which is the gated channels. These are gated channels present in the neuronal membrane. And what are they? There are gated channel, which is voltage gated sodium channel, voltage gated potassium channel. So the first one is potassium. You can see there are gates that can open and close as required for the potassium. And in here, there are gates present which will open and close according to the sodium ions availability. Whenever there is any change in membrane potential, potassium always tend to go from inside to outside, and sodium tends to come from outside to inside, and we will learn more about it. Okay, so in here, the next part that we need to understand is there are two gradients. What are these two gradients? One is called the chemical gradient, the other is called the electrical gradient. Chemical gradient is the one that causes the movement of these ions from a higher concentration to a lower concentration. Chemical gradient is very easy to understand. If you have, for example, a perfume bottle, you open it at the corner of a room, and you will see that you can smell it from the other corner of the room, because the concentration gradient will always move from a higher concentration to a lower concentration. Okay, so that's and that's true for ions in here. So wherever the concentration of one particular ion is higher, it will always try to move to a lower concentration. The other is the electrical gradient. We know opposite charge attract. Positive will try to go to the negative charge will try to go to the positive charge. So here we're talking about two gradients. One is electrical gradient, the other is chemical gradient. And we are talking about two ions in here. One is potassium ion, the other is sodium ion. So with the knowledge that we just learned so far, we now know that the potassium concentration inside the cell is higher. If you recap, because of the sodium potassium pump, the potassium ions is inside higher and the sodium is concentration outside higher. So let's take the first one, which is the potassium. So if you consider the chemical gradient, since the concentration of potassium inside is higher than outside, it will always try to move from inside to outside. Now let's consider the electrical gradient. Now potassium is positively charged and we know the inside of the cell is negative, outside of the cell is positive. So the potassium will try to move from outside to inside. Now, if you see the dimension of these two arrows, you will understand the chemical gradient is much higher or much more stronger than that of the electrical gradient. As a result, potassium always tends to move from inside to outside. So, at a point, if when the chemical gradient and the electrical gradient, there will be a time when both will become equal. So the point where the potassium chemical gradient and potassium electrical gradient become equal, this is known as the equilibrium potential. Okay, when both the potential, which is a chemical gradient, is equal to that of the electrical gradient, it is known as the equilibrium potential. Again, similarly, in case of sodium, we have the chemical gradient, which is the sodium is higher outside than inside. So the movement of ions will go from outside to inside in this case. But remember, the electrical gradient will also go from outside to inside because because sodium is positive, it will try to move towards the internal environment, which is negative. And this, both of them cumulatively, the electrochemical gradient, sodium, therefore always have a tendency to move from outside to inside. And when it reaches equilibrium, when the electrical gradient and the chemical gradient, at what point, of course, it will become equal, and that's known as the equilibrium potential. I want you to understand that this is positive 66 millivolt, and in case of potassium, that is negative 70 millivolt. We'll learn about equilibrium potential separately, but here, just to make you understand, when both these gradients, chemical gradient and electrical gradient, become equal, it's known as equilibrium potential. But for our current part, we need to understand one basic information, that potassium always has a tendency to move from inside of the cell to the outside, and sodium always has a tendency to move from outside to inside because of these gradient changes in the gradient. Okay, so now we are learning why does these sodium and potassium ions open. There has to be some sort of a stimulus that causes these changes. And the reason why this happens is because of these active channels. Okay, so we have three different active channels. One is chemically regulated channel, the other is voltage regulated channel, the third is mechanically regulated channel. The chemically regulated channels are with chemicals like neurotransmitters, which we will do again in a separate lecture. And whenever these ions, these chemicals attach to its receptor, they open these channels. As a result, sodium and potassium ions open and they do their action. They do the movement of these ions, which causes the action to take place. Or it can be voltage regulated channels. There is a change in voltage as we just learned. As a result, what happens? These channels open and causes the influx of these ions. The third one is mechanical regulated channel, the one that we learned about in the first slide when the person, stepped into the nail. That's a mechanical. They applied pressure that causes the channels to open, and as a result, the sodium and potassium ion movement take place. With that, we actually want to learn again what is the resting membrane potential is the negative ions along inside the cell membrane and positive ion along the outside. Okay, so inside negative ions, outside positive ions, and the potential energy difference is negative millivolt. The cell is always electrically charged and is called to be polarized. Resting potential exists because of the ionic concentration, differences in the ion concentration with extracellular fluid rich in sodium and the cytosol or intracellular fluid rich in potassium, phosphate, and amino acid. Membrane permeability differs between sodium and potassium. We have learned that, and we now know that the resting membrane potential is negative millivolt. Okay, so now we are heading into a different type, which is a third question. That is, these membrane potentials has happened, it is the membrane is at rest, which is the negative 70 millivolt. Now we are talking how to excite the neurons. The neuronal excitability, as I said, can take place by several methods. And here we are talking about neuron to neuron. This is one neuron. This is another neuron. This is the axon of the neuron. This is the synaptic, axonal cleft or axonal terminal. And this is the synaptic cleft where neurotransmitters are given in. And here in the dendrites, there can be ligand-gated ion channels that can take up these, neurotransmitters. They are, the ligand channels open. Let's put it again. These neurotransmitters, attach to their receptors. As a result, ligand-gated channels open. When these ligand-gated channels open, there is movement of ions. Okay. Now, if the movement of ions can take place in two ways. One is known as the graded potential. The other is known as the action potential. Okay. So, for example, only one sodium ion moves in, one potassium ion moves out. This is minimal, right? So this change is very local and it does not affect much. Stimulation of a neuron causes local disturbance in the membrane potential, and this difference is such that the membrane can become little excited, but the potential difference or the movement of these ions do not take place at very length. Okay, so the local potential, which is the change in the membrane potential, is graded, meaning that they vary in their magnitude or voltage according to the strength of the stimulus. So if the strength of the stimulus is not a lot, then what will happen it will go to a certain region and will die out. Okay, so let's understand this in the next picture. So if I have to make a graph, what is happening? This is the membrane potential. This is the time in which this is happening. Just give me 1 second. Yep. Um, and here, what has happening is small stimulus are coming. The membrane, so some of the sodium ions are moving in. As a result, what happens is from -17, which is the resting membrane potential, it goes into maybe negative 69, 68, 65, and so on. So, and then it dies down. Again, there can be another stimulus which is small, goes ahead and dies out. So these are very local stimulus. These are known as graded potential. These are graded potential. So they are local. The local potential are decremental, meaning they get weaker as they spread across from one point of origin. They decline in the strength, partly because of sodium leaks back to the cell through the channels along its part, and partly because as sodium spreads out over the plasma membrane and depolarizes it, potassium flows out and reverses the effect of the sodium inflow. What I mean is, I'm going to explain that in the next slide so that it makes sense. So you understand, this is the membrane, the potassium, there's positive side charge outside of the membrane. There are negative charges inside. Whenever there is a, stimulus, the channels open. You can see positive ion is moving in. And as a result, if the stimulus is not high enough, the magnitude is not high enough, no further potassium will come, or no further sodium will come in. As a result, it will dissipate out. That's number one. The other is, in the process, since it's slow, potassium will go out and it will balance out, and no further propagation of the impulse will take place. So it's short-lived, it's local change in the membrane potential. It decreases in intensity with distance, and sufficiently strong graded potential can initiate an action potential, which we'll do later. So if you can see in here, some potassium sodium ions are moving in. It is local. It changes the membrane potential locally, but it's not strong enough to go across the entire membrane. Okay. The local potentials are reversible, meaning if stimulation ceases, diffusion outside the cell quickly returns the membrane voltage to its resting potential, which is -70 millivolt. Local potential can be excitatory or inhibitory, like we have in other. We will talk about it in action potential. So some things to remember or recap is, whenever there is a stimulus, if the stimulus is not large enough, it will cause changes in the membrane potential. This membrane potential change is very local. It does not happen a lot. Not enough stimulus is there, that not strong enough, so that sodium which has moved in will eventually, leaks back into the extracellular fluid. There is another factor, potassium also can leak out. As such, the balance comes back to its neutral position, which is the resting membrane potential. So in here, we are talking about graded potential. And the stimuli, it can be mechanical stimulation, which is mechanical gated ion channels, which is through pressure. It can be chemical to a neurotransmitter. But remember, however this happens, it is local, it is short lived, and it does not go to a point of action potential. We'll understand this better through this figure. Like it's a very similar figure like the one that I just showed. So this you can see in here, graded potential. There is from -70, it has become -65. So some of the sodium ions have moved in, but there has to be another stimulus that has to come in order for more sodium to come out in, but it does not happen. As such, the membrane, the sodium ions either go back through the leak channels to the extracellular fluid, or potassium ions leak out. As a result, the membrane potential comes back to the -70 millivolt. Now, if you look in here, one important information to see in here is that these are dendrites, right? And these dendrites are receiving information from another neuron. It can be a pressure, it can be neurotransmitters, and the stimulus actually can be very local at one point and die out. Okay, so if I go back to the slide that we had started off, I want to show you these axons are sending neurotransmitters, and ligand channels are opening. They can be graded potential and die out, or it is possible there is another dendrite, another neuronal neurotransmitter release happens, and more neurotransmitters are released, and more ligand channels open. These graded potential can move to a certain point. At this point, they can come to a point which is known as axon hillock. These graded potential can sum up together to form or come to a region of axon hillock. And what happens in the axon hillock, it can lead to an action potential. Again, what is action potential? I'm going back one slide so that you understand it better. So, an action potential is a more dramatic change produced by the voltage gated ion channel in the plasma membrane. Okay, so here this resting potential, here graded potential together come into the action potential site, which is the axon hillock, which leads to action potential. So the question is, what is in the axon hillock that an action potential is created? Most of the soma, which is a cell body in here, has about 50 to 75 channels per square micrometers, not dense enough to generate action potential. It has sodium potassium channels, but not so many that it can create action potential. But the axon hillock, which is also known as the trigger zone, has about 350 to 500 channels per micrometer square, which is enough to produce action potential. If an excitatory local potential spreads along the way of the trigger zone and is still strong enough when it arrives, it can open channels and generate an action potential. So it can have excitatory impulse, it can get inhibitory impulse. If it gets excitatory impulse, it would lead to action potential. And if it is inhibitory, then it will die out. So we are here talking about axon hillock, which has tons of channels which can take up and cause the movement of sodium and potassium to a degree to create what is called action potential. So again, this slide, so we are talking about in this the ligand gated channels, which is opening up channels, which is causing graded potential. Now, summing up these graded potential near the axon hillock can cause action potential. There are tons of voltage gated channel in the axon hillock, which is the reason this is known as the trigger zone. So we are going back to the slide that we had started with. You remember the graph. This is the membrane potential. This is the time. Here we are talking about small impulses that are coming, that can be any chemical. It can be pressure, but if it is the stimulus is not strong enough, then it will go through a process and die out. It can be little higher, it can be little higher or little lower. But if from negative 70 it reaches to a point of negative 55, this part, which is shown in a purple dash, the negative 55, it is called threshold. This threshold means at this point, the action potential has been initiated. That means the cells are now, the sodium potassium has started moving in such a way that it can go long distances in order to create the electrical excitability to such a range that it can move from one part of the cell to the other part of the cell. This threshold potential is required in order for the action potential to take place. Now, here we are talking about any any neurotransmitter that is excitatory. We'll learn about excitatory and inhibitory neurotransmitter little later. Just remember, excitatory neurotransmitter like glutamate causes action potential to happen. And there can be inhibitory neurotransmitters like GABA, which can cause depolarize. It can cause it to go below the resting potential. Okay, so it can go below the resting potential and then come back to the resting potential later on. So this process, just look into this. Each of these waves, you will see there are three parts in here. One is this is going up, then a little bit of a plateau or a curve, and then goes down. Okay, so each one of them, whether it's graded, whether it's action potential, or whether the cell is hyperpolarized, the curve is pretty much the same. So what are these curves called? There are three steps in which this happens. One is called depolarization. The next is called repolarization. The third is called hyperpolarization. Depolarization, or the ray rising phase, is when from negative 70 it goes to 0 and then becomes positive, goes to negative 55, which is the threshold, and that phase from one phase to the negative 55 is the depolarization phase. And then when it reaches positive 30, from negative 55 to positive 50, then it tries to go down. At that point, it is known as repolarization, or is trying to return to its membrane potential. Negative 70, which is repolarization. And sometimes even it goes below the resting potential to negative 90 millivolt, which is known as hyperpolarization. So if you look in here, The stimulus, the cell is at resting phase. Stimulus comes, it reaches negative 55, action potential is generated. If it does not reach negative 55, it will be considered as graded potential and will die out. If it does goes into threshold negative 55, it will shoot up, which is the depolarization phase, then go into positive 30, which is the phase at which it will try to return to its resting phase, goes into repolarization and goes to the resting phase. At certain time, it can lead into hyperpolarization phase. So series of rapid occurring events that change and then restore back to the resting stage. Travels or spreads over the cell surface without dying out. So we are talking about this phase and again another phase, another phase, and another phase goes on continuously so that the impulse can travel from one distance to the other. And that's only possible if an action potential is created. Where is the action potential created? In the axon hillock.